Hello, everyone. For my biotechnology, I chose CRISPR, the gene editing technology, specifically Cas12a. CRISPR, what is it? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which are, in this image, you can see the repeat units are represented by the magenta. These were first discovered in 1987 inside of a bacteria known as Escherichia coli. Its discovery has led towards discoveries in capabilities in gene editing, as said before. CRISPR-based editing technologies are often useful for their ability to target specific DNA sequences and identifying them, as seen in this nucleotide sequence. Alongside CRISPR, there are associated proteins that make these systems, the most recognizable being Cas9, which is used to cleave sequences and to quote-unquote copy and paste. Cas12a is also an associated protein with CRISPR. Similarly to Cas9, Cas12a cleaves DNA sequences targeting genomic sites with CRISPR, and they also do the same things pretty much. Cas12a uses a special type of cleavage, which is known as cis cleavage and trans cleavage, which means that it cleaves a targeted strand, which is the cis, and an indiscriminate, which is the trans. As you can see in this picture, there's a wide cut here, which is one side and the other. There is many differences between Cas9 and Cas12a. Cas9 is a double-stranded break that is blunt, which means that there can be loss in functions. Well, on the other hand, Cas12a has this mentioned transcleavage breaks with the wide breaks, and they are staggered, leaving the sticky ends, which means that there is room for mutagenesis. And because of that, mutagenesis is when there can be mutations and the functions are retained. With Cas9, there are small base pair insertions and deletions, as I said, with cut and paste, while Cas12a has a wider range, which means that there is larger base pair insertions and deletions. For Cas9, to bind to the DNA sequence, there needs to be two RNAs known as CRRNA and tracker RNA. On the other hand, Cas12a has a single CRRNA. And with Cas12, it's special because it does not need the promoters that Cas9 needs, and it processes its own CRRNA. With Cas12a CRISPR, there are benefits that can be brought from this gene editing technology. It has the larger insertions and deletions that were mentioned previously. It is a good material for investigating the loss of function phenotypes, such as the ones that were mentioned from the Cas9. It also has the high efficiency because of its cut size and its accuracy. It is useful because it processes its CRNA, just as I had said before, and it also can be used. It's effective for knockouts and knock-ins, which will be explained later. A knockout and a knock-in, what is that? A knockout is when something is removed from a DNA sequence. A sequence becomes inactive and is disrupted, while a knock-in is an insertion. The insertion is considered losing one function while replacing it with another sequence. Here you can see a knockout where there is a disruption here. And then with a knock-in, you can observe the insertion with something new. In the past, CRISPR systems have always been used to target DNA sequences. Over time, more technological advances have been made to make more accurate and efficient additions to this. For example, CRISPR-Cas9 has been developed to knock out microRNAs, which are used to control expression in genes. And in recent years, it's been proven to have smaller insertions and deletions. On the other hand, CRISPR-Cas12a has been improving upon this by having its wider range and its larger insertions and deletions. In recent studies, there was to test the capabilities of CRISPR-Cas12a, a study had explored its targeting abilities of the system. From this study on mice and imaging, it was found that the microRNA imaging system can identify tumor cells in the glioma, which is a area of the tumor. In another study on these gliomas, the researchers had targeted a microRNA known as MIR21, the driver of growth in tumors. By disrupting this MIR21, it was found out that the knockout of the MIR21 in the tumors had caused reduced growth in the tumors, as well as an increased survival of the subject, such as rats, of course, in the cells that they had taken. In the future, there can be much progress and there's hope for developments in the CRISPR systems. For example, CRISPR-based therapeutics for human gliomas could actually be something that we can have in our reality, as discussed in the tests from before, although they have not passed clinical trials yet, of course. There could be more research on CRISPR cancer targeting, such as the miRNA therapy, and passing the clinical trials. There could be research branching out into other fields other than what has been discussed, such as cell biology, neuroscience, and cancer biology. There could be more advancements made in the screening and imaging that I had discussed with molecular diagnostics and screening. Although that there is hope for this, there is also still limitations and challenges. Although widespread, CRISPR-Cas12a still has the off-target effects that other CRISPR systems have, which can lead to unintended mutations or disorders, which is not a good thing at all for things, even if we're trying to get it towards humans. Another problem is finding the proper plasmid that can deliver these systems to the sequence. And there's always like drawbacks that can be found in this. The problem is finding the proper one that won't have as much low delivery efficiency. Another problem is that there isn't enough research on the cancer targeting and the targeted imaging to pass clinical trials, as said before. And another problem is that CRISPR-Cas12a requires a specific PAM sequence, which is a sequence used for binding to the sequence and recognizing, which is known as the TTTN. And because of this, the sequence isn't as versatile as it should be. So Cas9 is sometimes still seen as better than Cas12a. With ethical problems, there are still things that come into question, especially with gene editing. The main problem is always considering the health of who is being treated. Unintended outcomes such as the off-target effects can be seen as risky for humans and can even like be health it can be risky for health. And the adverse effects are always the biggest problem with something like gene editing. Because of this, in the future, it's always important to improve upon what is safe for each type of gene editing, specifically with how precise this is going to be. And that is all. Thank you very much.